but first we will go through data types you will probably face with, how to handle them, how to read them or import them into R, or I think this is also quite universal. So any kind of programming language you use, it's a kind of the same way. And then finally, I managed to understand the dplyr and the other things. So I will give you a short guidance on that as well, not too much. And then I would like to discuss much more like general things on programming at the end. So because we are doing bioinformatics and we are dealing with biological data, the main data types we had to face with are mostly tabular data. Many of our programs outputs are tabular data, like Excel tables. And also we have to deal with a lot of strings, text-based data, like sequences. And we know how to, and we have to know how to handle these things. I found another very good cheat sheet, but I haven't sent that because this is not the only one, or not the only thing I would like to send to you. But there is actually a website for cheat sheets, RStudio cheat sheets. What I will do now, I will just copy the link and put it into the chat window, and then you will have it later. Many good things, and this will be one of the good approaches, I think, when you start building up your own scripts. So it will be on the chat window later on. It's actually a very simple link, a lot of things. So if you look at here, there are so many, also deep learning. I don't think many of you will start programming deep learning as the first program, but there are so many other things as well, which is very, very useful, especially if you learn how to use this. I haven't used too much of these earlier, but now I'm becoming more and more familiar with these. But of course, it's not enough just to use these. I will tell you what is my preferred approach later. Okay, so if we are speaking about data importing, or how to read in your data into R, there are two fundamental ways to do this. The first way is to read in everything, what you want to process or what you want to work with. And this is usually some kind of tabular data, what you would like to handle as a whole, data on your parameters or outputs or results, what you would like to plot and visualize. And also there is usually a limiting factor what you can do, which is your memory, the computer memory. So if your table is huge, then it's probably simply just bigger than what you can read in one go. So if it's like that, then you have to think about A, go to a machine which has more memory, or B, do some kind of pre-processing on your data, make it smaller, and then read it in. So if you read in your file, then there is this tabular data reading command group. It depends on what kind of data you have. So if you have a CSV file, a CSV means that the fields are separated from each other by comma or semicolon sometimes. Then you can use the read CSV or read CSV2. If you have a tabulator separated or space separated data, then the read delim or read delim2 is the function you would use. We did it many times already, so much of our previous examples, when we read in data file, we used the read delim function. Again, there is one thing what you can see on the cheat sheet. So you can see, okay, if I have a file with tabulators, then I should use the read delim or read table. But every time if you want to, oh, okay, I heard something from someone. Anytime if you want to do something a bit more sophisticated reading, many of these functions, for example, can skip a few lines. There are so many programs that are actually has an output of some kind of tabular data, but before the main table, you have something else, like a few lines of headers, a few lines about how this data was generated, who generated the data, and you don't really want to read that into your data table or data frame. So in that case, just go on the help, put in read delim, and you will see that there are parameters within the function like skip, which means that you can skip X number of lines before reading in the main table. There is a special case, which is quite rare nowadays, but it was very common like 20, 25, 30 years ago. And there are some databases or some websites where you can find these kind of data formats. The most infamous one, I think, is the PDB. The PDB is the database for protein structures. The main PDB format, even nowadays, is not space, not comma, not tab separated, but it is it has everything in its place by a position. I think there is a this read FWF can handle files like this. This is very dangerous to handle files like this. The reason I just write down to you how it can look like. So in PDB, for example, the atom coordinates, all the atoms have their own coordinates in a three-dimensional space. That's how you can visualize a protein structure. They have an atom line, which looks like atom and starts with a word atom, and then something else, probably the number of the atom and then the number of the residue, something like this. And then the type of the atom, which can be just an N if it's nitrogen, or can be a Ca if it's the alpha carbon. But as soon as it is something on a amino acid side chain, this field can be longer than what you would expect. So if it's just a Ca and then you have your coordinates, let me just put in 3 coordinates like this, which is the XYZ coordinate. And in this case, you can use space, or actually you can use something which is called white space. Almost all programming languages can handle white space. White space means that it's 1 space or 2 spaces or 3 spaces or 2 spaces plus a tab. So everything which looks like a hole, like a gap, is called a white space, and usually this is just handled as one delimiter, the same way how this is handled as a one space as a delimiter. But in this format, if you have something where you have your atom description, it looks like C gamma oxygen H, which is one of the hydrogens on that oxygen, then you can see there is no space anymore. So you cannot use the space or white space or anything else as a delimiter, just because PDB always puts the coordinates into the same position, which is, I don't know, let's say the 15th or 21st position, 21st position. So this is what you have to be very careful when you do this. I think the the gene bank header has a kind of problem like this as well. So I think the very first line of the gene bank format has some kind of limit of characters as well. Even if you use too many characters there as the name of your sequence, I think, then it can merge with the next field, which is probably the size or something like that. So there isn't too many cases like that, but there are a few. So always be very careful. Always read the standards of your input format and read in accordingly. Also, it's very important to make differences sometimes between white spaces and white spaces. If you use tabulators as the delimiters between fields, then in theory, you can use spaces within the fields. So for example, if you have something like a FOSTA header, a FOSTA ID from a UniProt record, there are quite a lot of spaces within the FOSTA header, and they should be in one record because they are provide that the ID line for that sequence as a whole. So later you can just put this into your tab separated table, and then the tab will be different from the space, so it won't be handled as a white space, but you'd have to specifically use the tab and not just anything or just simply white space. R is a bit better in this from this perspective, because first of all, you usually see what you are importing if you are using the import dataset function in RStudio. So you can see, oh, there is something wrong because something which I wanted to put into one field was actually split, and it's now in 4 or 5 different fields. But when we will work in the command line environment, different programs are using different default delimiters. So it's always very important to know what you have to use. If you cannot use tab, because, for example, you have to put tabulators in your data, in your text-based data, then you should think about using comma or semicolon. Comma is quite often used, so I think it's better to avoid the traditional CSV, which is looking something like this. If you want to use some kind of comma, I would much more use the semicolon as a separator. I think it's much more safe. It's much rare that you use semicolon in your data. It's also important to know that in many countries, the comma is used for decimals. For example, in Hungary, if you are saying 2.5, then it is 2.5 in Hungary and not 2.5. So it's better to avoid, I think, comma as a field separator. Much better to use semicolon. Okay, so this is approach A, when we read in the whole table, and then we will process it and visualize it and filter and subset. And this is what we will speak about later. But there's another way how you can also read in data, and this is especially important, I think, in bioinformatics, when you are working with huge files, which you just simply cannot read into your memory. Think about a FASTQ file, gigabytes, hundreds of gigabytes sometimes, but you want to do something on these files. So in this case, you can do it line by line, or you can do it in smaller chunks. Today is a special day because I actually practiced this before. So if you don't mind, I will just copy here one of my previous small script parts, and I will tell you what I'm doing here. You won't find this FASTQ file anywhere. I didn't put it up, but it's the same for every FASTQ files. If you are not familiar with the FASTQ format, then of course we can just Google it, and I can show you what's the specialty with the FASTQ. The FASTQ is usually how you get back your results from next generation sequencing, and one record consists of 4 lines. Always one record consists of 4 lines, from which the first line is the ID. Usually this ID is more descriptive, something like this, for example, where you have sometimes a date, an identifier for the chip which ran your sample, and usually you also have a position. The position is actually very important. What is the coordinate of your sequence read? So these things are, happening in a 2-dimensional space, on a 2-dimensional surface, and the sequencer actually detects color changes in a matrix format. And the reason why it's very important to know where is your sequence read is if there is something wrong with the sequencing right next or in the surroundings of this read. So for example, the sequencing is not working because the DNA was damaged there. Then there is no light nearby, and the light of this spot can be bright enough to go to the neighbor spot as well. So you will have a duplication from the same sequencing, which is, if you just sequencing a genome, it's not a problem, because then you will have one more read for that particular part of your reference genome or your genome of interest. But if you are doing something quantitatively, like RNA-seq or microRNA sequencing, then it is very important how many sequencing reads you have from one gene from one microRNA. So that's why this is important, and that's why when you are doing your deduplication steps, what we will do when we will learn about how to pre-process next generation sequencing data, during the deduplication, the program is looking for positions as well. So this is the first line of the FASTQ file, and then you have the sequence, which was read. Then you have a comment line, which is usually not used for anything, and then you have a line which contains the quality score for each nucleotide read. So it's always the same length as your sequence, and the first character is referring to the first nucleotide and so on. So this star is the quality score of this G. The reason why this is not a simple 0 to 9 scale, because they wanted a more detailed scale, but you cannot use, of course, 10, 11, and 12, because then it will be 2 digits. So you won't be able to assign the digits to the nucleotide that easily. So in this case, it's always the same position. So what you have here now is actually an Ascii table scale. I think it's somewhere down here on the Wikipedia page as well. We usually use the thread 33, so this red one. So the exclamation mark is the lowest quality, while the capital H and I, GHI, these are the highest quality scores. So this is the range which is covered by this thread score table. So if you see something which is a bit more like just signs or even numbers, this is not as good as if you see letters. And there is something which is very important here. As you can see, the @ sign can be a quality score. Why this is important? Because you just simply cannot use the @ sign as the identifier line. So if you remember in FOSTA files, you always have that arrow or FOSTA ID line, this one, at the beginning of your ID line, and never you have it in your sequence. So this is a very discriminative sign in a FOSTA file. In FOSQ files, you cannot say that, okay, let's find all the lines which are starting with an @ sign, and all of them will be a sequence ID line, just because it's very easy that it can happen that the same kind of @ sign will be here as a quality score in your quality score line as well. Okay, so if you are dealing with a FOSQ file, you always have to deal with the line numbers. You are reading in 4 lines in one go, and you are doing something with the first line, you are doing something with the second line, the third, and the fourth one. So in like a very simple example, if you would like, for example, if you would like to do quality filtering on your data, then what you do, you are reading in 4 lines from your FOSQ file, you are checking the average quality score, which is the translation of these signs to numbers, and then a simple averaging. And if it is above a certain threshold, what you decide to use, then you print out all 4 lines into your output file. If it's below, then you just throw away all 4 lines. So how do you do this in R? So first of all, you have to, of course, define where is your FOSQ file, and this is the first line what you see here. So you define a variable which will be just an object. It's not, the file command doesn't read in the file. It's just like the address in the memory. So from now, the R program and the computer will know that this object is actually an arrow, a pointer to a file in somewhere in your file system. And you also have to define what you want to do with the file. If you say open equals to R, that means that you are just opening it to reading, so you won't modify the file. And then you have to do something, and I will explain this next, but also at the very end, it's also very good practice to close the file. You don't have to, because as soon as your R script finishes running, these 5 pointers are automatically closed. But the reason why it is always better to close here, because if you reopen the file and you do something else on the same file within the same script, if it wasn't closed and you open again with this line, it won't go back to the beginning of the file. It will go to that place where it was finished, where you finish processing it. So if you go along the whole file, then the 5 pointer will point to the end of the file. So you cannot really do anything again on it. So what am I doing here? The main command you have to know if you want to read in files line by line is scan. So scan gets the 5 pointer as the first argument. So this is our con, which is the connector or the object which is the 5 pointer. You have to say what kind of data you want to read in, because this is a text-based data. The what is equal to character. Otherwise, I think the default, again, if you look at the help of the scan, it's in the base packages anyway, so it's always there in your R program. So the default, as you can see here, is double. If I don't give this what equals to character for a FOSQ file, it will drop an error to me, because double is a kind of number type, and it doesn't find any numbers. It will start with some at blah 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 blah, and it will give an error. And then you are giving how many lines you want to read in. And we are reading in 4 lines together. The quiet is just covered. There isn't, yeah, just to make it a bit less. Yeah, I think it's, if it's false, can we print a line? Yeah, I think it's just asking scan not to report how many lines were read. That's the quiet. And then the separator, again, very important, because the separator in default for scan is white space. This means white space. So that means that every space is, after every space, it will go into different cell. Scan is always giving back a vector. In this case, because we are reading in 4 lines and we are using the new line character as a separator, we will have a vector with 4 elements, which is exactly the 4 different lines. So we will have a vector where the first index 1 will be the ID, index 2 will be the sequence, index 3 is the comment line, and index 4 is the quality line. So this is what the scan does here. So what it gives back will be pumped into the TMP, which is a temporary vector variable. This will be that 4-element variable. And we are checking the length of this variable. Why do we do this? We know that it has to be 4. Why do I want to check if the length of a 4 element vector is more than 0? While I know that it has to be 4. The reason is that when scan reads in 4 lines, it leaves the pointer at the end of that 4 lines. So if you ask scan to do this again, it won't scan the same 4 lines. It will scan the next 4 lines. It will scan the next 4, the next 4, and the next 4 lines. This is why this is in a while loop. And what is happening? It will read in that 4 lines into TMP variable. We will see that TMP variable within our while cycle or while loop. In each cycle, we will have 4 lines in our TMP variable. In each cycle, we will have a new 4 lines. And where there is no more 4 lines left, just because you reach the end of the file, then the next scan will give back a 0, an empty vector. And the length of the empty vector will be 0. It won't be more than 0. So as soon as scan reaches the end of the file, this while loop will finish and exit. So you don't have to worry about. This is one of the most common uses of while loop. If you don't know how much cycles do you have to do, because you don't know how big is your FASTQ file, sometimes it has 1 million records, sometimes it has just 10 records. But while will always go up until it ends. And if we just simply print out this, the TMP in every cycle, and then also I print HOHO just to see where we are, and I run this. And as you can see, this is one of the records. This is probably the last record, having index 1, 2, 3, and 4 here. So you can do this with any of your FASTQ files, of course. First line, an ID line. Second is a sequence line. The third is a comment line, and the fourth one. And the HOHO is always there as an extra, because we printed out it in every while loop. Okay, this is how you can do. Of course, if you have a read pair, usually a parent read, then you can do another scan within your while loop. So you are scanning your R1 file, which is your one of your sequencing pairs, and within the while loop, you are also scanning in the other one, R2, from your other file. Of course, you will need con1 and con2, so 2 5 pointers, of course. They will go together, and the reason why you have to read in both records at the same time, because usually if you want to filter, for example, for quality, then you want to have both reads above a certain quality threshold together, and you may throw away one read even if it's good, just because the pair is not good enough. Okay, the next thing I will show you is how to, is everything all right and clear with the scan, and how can you process this? I hope so. The next thing I will show you is how to read in a FASTA file. I also, I spent quite a lot of time to figure this out. And one of the reasons is because I never do this in R again, but it's much easier in Perl. We are starting the same way. So our aim here is not to process this file line by line, but read in the whole multi-FASTA file. So this is actually a MAC-A gene or MAC-A protein, which is an antibiotic resistance gene collection from the UniProt. I just downloaded. Again, it's not important what is this, so you can do this on any FASTA file. You can just download from the UniProt, and this is not just one protein. So this is a collection of, I don't know, like 100 proteins almost. So what you have in this file, I can show you, I think. Actually here. So it looks like a very standard FASTA file, sorry, a FASTA ID line and then sequences, FASTA ID line, sequences, and so on. And of course, while I want to show this to you is because one of the problems we already spoke about last time, if you want to look for a sequence pattern, and the sequence pattern starts here and goes here, then you cannot use a simple sequence or character search because there is a new line between them. So what I will do is I will read in this whole FASTA file into a data frame where we only have two columns. The first column always contains the whole ID line, and the second column contains the whole sequence without new lines, without spaces, so as a continuous string. This is, again, something what you will probably do very often. So here so this is the file connector, and this is a data frame, an empty data frame, where I say I would like to make a data frame with two columns. The first one has a column header ID, and it will be a character type. And the second one will be the sequence, SAQ, which will be a character type as well. And this is very important. I spent quite a lot of time while I figured out that that was the problem. R is always tried to transform strings to factors as soon as, even if you say that I want to use this as a character type, I want to do this as a character type. If you start filling up your data into this data frame, it will be converted to factor. But if you use this argument here, strings as factors equals to false, so please don't convert my strings to factors, then it will remain string or character type. I also initiated a counter, which is 0 in the beginning. The while loop is very similar to the one we had before. So it starts here and ends here. And of course, we are closing the file connector here. You can easily use the same variable here and here again, because you are just redefining what is con. You defined con here, closed, used the con here, closed con here, redefine con here, use it here, close it here. Of course, you shouldn't redefine it somewhere inside. So what is happening here? We have the same scan, almost exactly the same as we had here. There is only one difference, that we are reading in only one line at a time. And we are deciding what to do with that one line. So this one line will go into TMP again, but it will be actually the first element of our TMP vector. So this is what exactly contains our line from the file. So we have to decide what to do with the line. There are two possibilities. It's an ID line or it's a sequence line. If it's an ID line, it has to start with our ID identifier. So this is from our last meeting. How do you detect patterns in a string? str underline detect. This is coming from the string R package. And this will give you back, let's just bring up the that cheat sheet. So I'm using this str detect because what I want to get is only a true or false. Because if you are using if, you are only want to know if this is true or false. So it will be true if the line contains this sign. In this case, you can just use TMP, but you can also use TMP1 like this. If it contains this sign, then do this. If it's not having this sign in the beginning, do this. So this will be done if it's an ID line. This will be done if it's a sequence line. So what do we do? First of all, because we started the counter with 0, we will increase the value of the counter by 1. This will be our row identifier. So our count, this C and T variable, I will use it to define that, okay, I want to put this data into a new row, and this is why it is always increasing by 1 as soon as I reach a new ID line. And this is our data frame, which we initialized here. And I'm saying that, okay, let's take our data frame and put into this row and this column. If there is nothing here at the column part, it means that we are putting things into all columns. But this is the row identifier. And put in the TMP1, which is our ID line, into the first column, which was the ID, as we defined here. So ID will be equal to TMP1, and the sequence will be equal to nothing. But at least I initialized this variable. I cannot do it without it, because then, because the data frame is always, in data frames, you always have something in every cells, in every columns. So that's why I had to put in at least an empty string. So this will happen if I'm in an ID line. And then I go to a sequence line where I already luckily get rid of the new line character because I used it as a separator by scan. So the scan already trimmed or ate the new line character from the end of the line. So what I want to do is just simply, I want to put into the sequence column of the counter row my sequence. And why do I do this here? The paste, I think we already had the paste before. The paste is just gluing strings together. I will glue this string to this string and use separator as nothing. The reason why I have to define this here, because if you look at the command paste, then you will see that in default, the separator is a space. So if I don't use nothing between two quotation marks, then it will put spaces instead of new lines into my sequence, which I don't want. So what I'm doing here, I'm taking the value of the sequence, which is empty or which is already started filling up, and I'm putting my actual sequence line, gluing them together and put it back to the same place. So what will happen with this FOSTA file? It will read in the first line, put into the first column, first row, first column, ID line, ID. Then read in the second line. It realizes that it's a sequence line because there is no ID line identifier in the beginning. So it will put it into the second column where was an empty string. So it will merge this to an empty string. So this will be a new string without new line, without anything else. It goes to the next line. It's again a sequence line. And it will paste this one together with this one and put it back to the sequence field and goes up to here. This will be the last, which will be merged together, and the whole sequence will be in one string. So if I'm running this, and then I'm looking at the df variable, which is here, then you can see that we are having our ID lines in our ID column, and we are having our sequences in the second one. And from this moment, you can look for your short linear motifs if you want within the sequence and put them into a new column, any kind of pattern. If this is not a protein sequence, that a nucleotide sequence, this is what, for example, we did as a short project yesterday with Ibrahim, then you can just extract nucleotide positions easily from these sequences with the string manipulation tools. Right, so these are the two main types I wanted to cover before we go forward, the FASTQ file and FOSTA file. But if from this moment, I think if you have any kind of files you have to read in differently, not the usual way, not as a table, you can just use these two examples and rewrite them as you want. There is also, it's also quite common that you have a file, an output file, where you have those kind of headers I mentioned before, where you have something before your real data, but you don't know how many lines you have before, because it depending on, it can depend on many factors. It's also a good way then, if you do the same kind of line by line reading, and you define a variable before that, saying that, for example, pass equals to 0, and you only change that pass variable when you reach that line where your data starts, and you are starting reading in and processing the data from that particular line only. So actually what you did with the first few lines is you read them in, but you didn't do anything with them, up to a point where you check with an str detect that you already have that string, you know that it's the start of your real data, and from that moment you are reading in and processing the data as well. Okay, so we are going forward to the data manipulation, and I want go through all the commands, because I think, again, if you want to do anything, any kind of data wrangling, then just please use these cheat sheets, and then always go into the more detailed description of the function. This is what I will explain later. But now we have to include these few libraries. Maybe not all of them are installed for you to use, so I will just wait for a few seconds or minutes. The dplyr should be there, the ggplot is there, the reshape2 is there. We already used these free in our previous meetings, but maybe this TIDYR is not there. So just please, if you don't have it, just as the usual way, just go on the packages tab here and install package, and just open TIDYR and click install. I don't install because I have it. So I will just wait. I will just go for a glass of water while everyone finishes all the installation. If not, then we are going forward. So just highlight these 4 lines, Command Enter, and include these libraries. I will make a bit of space here. Again, I practiced this before. So what we will do, we are going back to this infamous replica exchange data set. I think I will copy this whole thing and I will go through step by step. So as you may remember, this RxDF, which you should already have, this RxTemp txt file, or which we used before. This is the output of a replica exchange simulation, and this is the temperature output of 8 different structures. So if I read this in, and if I look for the data, it looks like this, where we have a time step in the very first column, and then we have numbers here, which are actually relative temperature numbers, or relative temperature values for all the 8 different structures we are simulating in this replica exchange protein dynamic simulations. And what we want to see in this file is how the different structures went, so where they were, if they were stuck into a specific temperature, or they were stepping up and down normally as we would like. There was also a question before from Jinji, if we can put more than one data set onto one graph. So I will show you that it is possible. So first of all, this next line is just simply gives a bit more descriptive column identifiers. This is what you should always do. So if you don't have your header in your table, in your txt file you are reading in, then please don't leave v1, v2, v3 up to v9, because of course I know what is this now. Maybe I will know 1 week later, but I'm sure that I won't know what are these things in a half year or a year later. So if you really want to keep your scripts and codes tidy, then please give names for the columns, and this is how you of course give. This is what we did before already. So this is just giving the names for the different columns. So if I only want to visualize one structure, then it's not a problem. Then this kind of data structure is perfect. So as you remember, if you say ggplot, you give the data frame you want to work on, and you then give the x-axis and the y-axis, and you just want to make a simple line graph. Then it looks like this. I didn't run this line, so it looks like this. So what you can see here, that this structure went through quite a lot of different temperature stages. Then it's stuck actually between the 2 lower temperatures, sometimes going up to the third lower temperature box, which is good or bad. It's not important at the moment. It has quite a lot of steps. That's why it is quite dense, this graph. So if I want to put on structure 2 on the same graph, then if the data frame looks like this, then the only way how you can do it with ggplot is actually to introduce a new data and a new graph. So this is actually the answer to Gingy's previous question. If you can put multiple data into one plot, yes. I put the same thing here, but it's only because I want to plot another structure on the same. But as you can see, it's the same up to here. So this will plot the first structure, and then I'm doing another geom line. I give a new data, which is actually the same, but it can be anything else. And I'm also defining the x and y axis. The x-axis, of course, the same as was before. And just to make it a bit different, I will color it to red. So if I run this line, then you can see now that we have two data sets on the same diagram. And we can also see how this structure went through the different temperature boxes during the simulation. Maybe you remember that we also did a violin plot to visualize where these structures spend their time. So if I'm only to this first, so this is almost exactly the same what we had before, but this is a violin plot, so we don't have x and y axis. X is only, in this case, just a string, a factor. So if I'm plotting a single structure, it looks like this. Again, the violin plot shows you that this structure spent most of the time in this stage, a bit less in this stage, and that was the third one, and the fourth one was this, and the sixth one was this, where it spent most of the time. But in the same way how we did before, I can just add another geom violin, defining its own data frame and defining its own x and y coordinates and a color. So if I'm running this whole line now, then you will have two different data sets on the same graph. Again, it can come from two different data frames if you want. In my case, it came from the same data frames. Of course, you can see, yeah, I just put here this extra line to show you how this actually, how the structure builds up. So you always start with ggplot, but you can just start like this, saying that, okay, I am starting doing a plot. It's like taking out a paper, a scrap paper from your drawer to draw something, to sketch something on the other side of the paper. Okay, and then I will add this to the paper, and then I will add this to the paper. And this is exactly the same as the line before. So I'm now defining the data for the first plotted thing here, and the XY axis here, and the fill here. And I'm defining the second data here, and all these here. So if I'm running, it's the same. Maybe it's just colored. No, it's the same color as well. Of course, as we saw before, we have 8 structures here. So, Kota, your microphone, I think, is on. I see that your question is, don't know how. It's okay. It's much better now.
Darn it.
Thanks. Okay, so there is a question from Kota. Actually, don't know how to automatically pick scale for object of type formula. Defaulting to continuous error aesthetics must be valid data columns. This is what you get back as an error message, I think. I think the problem is that there is an underline in here. Also, if you have it here, I think this can be anything. I guess this is just gives you what will be written here down on your plot. Yeah, but this is actually the name of your column. So if you named your column like this, then it has to have the underline here as well. So if I say this, then it's just object not found. I don't know if this is the problem, or probably there is some kind of mistype, or you didn't put any double quotes for your X. Okay, we can go back to your problem. Okay, that was the problem. Thank you. Right. So if you want to plot all 8 structures on the same graph, then you can do this. I mean, you can just say, let's copy this line 6 more times and write 3, 3, 4, 4, 5, 5, 6, 6, and so on here. And that's what you can do with ggplot. But of course, you are writing a lot of things just instead of solving the problem in an easier way. ggplot cannot do it simpler with this data frame. And this is why we modify this data frame. And this is how we will modify this data frame again, but in 2 different ways. Actually, I think in 3 or 4 different ways. The first is melt. This is what we did before. This is it. So melt is a command from the reshape package, reshape 2, as far as I remember, but I'm checking it. Oh, this chat window is always on the way. Okay, so if I'm saying melt, yes, it's coming from the reshape 2 package. And this is, I think, both reshape 2 and ggplot, and I think also this dplyr is coming from the same kind of person or group. So this is why they are, of course, preferring the same kind of data structure. So what melt is doing, if you are giving an ID column, which is our time step column, then it will be untouched. It won't be changed, but everything else will be put in to a new column. So what will happen? It will happen that, okay, I know that I have a lot of data for structure 1 for all these time steps. Let's say that I have time step 0, and for structure 1, I have the value 0.45. So let's reduce the whole big table into 3 columns. The first column will be time step for that particular structure and for that particular temperature. And second column will be the structure identifier, and the third column will be the temperature itself. It will be, there will be a lot of redundancy in this table, of course. So it's, if you think about it, this is the most condensed way how you can store data. And I don't want to convince you to store the data in any other kind of format. This is the best way to store data. This is the way how you already do in Excel. This is perfectly good for having the data in long term, but it's not good for ggplot. So for ggplot, you will make a much bigger variable with much more lines and much more redundancy in it. So what happens if we run this mad command. Okay, I just run ID because that was highlighted. I wanted to run the whole line. Okay, so we will have this Rx underline df underline gg variable in our environment. If I'm looking at into, then we have time step column here. We have the variable column, which will be structure 1, and its value is this. And if we go down a lot, then after a while we will meet with structure 2. Please don't do it in your computer. It's enough if I'm just doing this. I think it was like 400 something lines. So what I just want to show you is how redundant the whole data frame now. Oh yeah, it starts somewhere here. So it's exactly the same. It will count from 0 up to 378,500. Here again, the only change is that it has structure 2 written into the second column. And of course, the temperature value is reflecting to structure 2. And this is the same for structure 3, 4, 5, 6, 7, and 8 in this table later on. It's again good to use a bit more descriptive column headers. So the time step remains time step, but a variable and value is maybe not the most descriptive. So I just modify the column names here, and we'll give time step, structure ID, and temperature as the column names. So if I'm going back to my column, it's now much more descriptive. And then it's way much easier to plot this. You just say ggplot. I want to use this rx underline df underline gg. So this is now the ggplot formatted version. I want to put the structure ID. So we are doing the same kind of violin plot we did before. So this one, but in one step for all the 8 structures, okay? So on the axis, we will have our structure ID, column. On the Y, we will have temperature. We are losing the time step at the moment, but we don't really care about the time. We just want to see where was the structure in terms of temperature. Okay, and we want to make a geom violin. And it's here. All of them are here. If you want to color them, then you just simply say for the GM violin that you want to fill them by structure ID. I'm not sure if it's working like this. No, I think we have to put this here. Yeah, and it's now different in colors as well. And of course, if you want to define a different palette, a different color palette, it's not so bad. This is the default color palette for R or RStudio. But if you want to use the one of the color brewers one, you can also use it directly from here. I just found it out. So if you want to say scale, fill, manual, it's important that I'm using scale fill manual, not scale color manual here. If you are doing a scatter plot, you are coloring the dots. If you are using something which has a inside, if it's like a bar graph, a column graph, or a violin plot like this, then you are filling it in. You can also color these plots, but then you will color the outline, which is now remain black for all of them. So what I am redefining is how will I fill them. And instead of using manual, I'm using scale fill brewer. And this is the same kind of things what you should find, what you can find on the Color Brewer website. Yeah, it was here. I showed you before, colorbrewer.org, colorbrewer.2.org. So if you want to use, for example, the quantitative scale and this one, so this is also, this is a qualitative, the second one, the number 2 palette, then you just simply say, other way, that I want to use, let me just check what is the way to define scale fill brewer. So you have to define your type, which is, if you go down here, the type is the sequential, diverging, or qualitative. So this is the same what you can see here, sequential, diverging, or qualitative on the website. So I will use the qualitative type equals to, and I don't have to, I just have to write qual, Q-U-A-L. I want to use the palette equals to 2, which is the second one. If I'm doing this, I will have a different palette. No, it is the one. So it is like this greenish, gold, blue, and pink. Yeah, green, gold, blue, and pink. So this is that color palette. Exactly the same what we have there. Right, so this was one of the ways how you can reshape your data frame. But what if you don't like using melt, but you want to use the plier package? You can. You will use the gather function. So for running this, you have to have the dplyr. If you look for the help of gather, then you can see that this is the part of, oh no, so it's the TIDYR package. But this is also something that I asked you to install previously. So what you have to do, again, you are just giving the original data frame. This was the Excel-like data frame. And you are giving those columns what you want to gather, what you want to put into separate columns. And then you will give your key and value description. So in one step, you are doing with what we did with math in 2 steps. So in math, we told the math what we want to keep, which column we want to keep and not melting. And then we defined the column identifiers. Here we are defining what we want to gather. I will tell you another way, which is much more, which is a lazy way and more simpler way. But we can also define here our column identifiers structure, which was structure ID here. Here it's structure, and temperature as a value. So if I'm running this and going to this one, we have exactly the same structure, same table structure as we had. Instead, we have structure here, not structure ID, but it's okay. You can, of course, rewrite this anytime. So in this first line, I define one by one that I want to gather all these columns. But what if you have like hundreds of columns and you want all of them to gather? If you have, for example, more than 8 structures in the same kind of experiment, if you are doing it with 24 structures, you don't really want to write structure 1, 2, 3, 4, 5 up to 24 here. So what you can do with gather, you can also define what you don't want to gather and put together and transform. And this is why there is a minus sign before this. So what I'm doing here, I'm gathering everything except time step. Okay, so if I run this, it will be exactly the same. It looks exactly the same. If for some reason you want to preserve two columns, which in the case of this data, it is a stupid choice, but I will show you because this is how you can see what is happening with this command. But of course, we cannot use this data frame to anything, but let's just run this. So in this case, I asked the function gather to gather everything except time step and except structure one. So how it looks like now, you have the time step column, you have structure one column, which was the same as we had here. So only the temperature values of structure one. And then you have the structure identifier for structure one you don't have because it was not merged. And then you have the temperature values. In this case, maybe if we want to make a scatter plot on the different temperatures for structure one and structure two, it will give us a chance, but there is no real point to do this. So let's just go back to the previous one because I think we will have a few extra ggplots here for this one. And maybe also some filtering steps. Let's see what I prepared for you. Oh no, okay, so this is what we will do now. And I will copy again the whole thing and I will go through all the steps one by one. So we are going back to our supplementary data table. This was the table which we again already read in before. This is the one of our metagenomics experiment final data table. Let me just read in and I will show you what is this. I will leave you some time just to copy this line. I just realized that it's much easier for me to write in the lines because they are already written. So it will be put into this SD data frame. And if we look at this SD data frame, this is a huge data frame. So we have quite a lot of columns and also 6,164 lines or entries. So this table had a header. So that's why instead of saying read delim header equals to false, how we did before when we didn't have a header line, we just use the default true. So we don't have to say here comma, but we don't have to header equals true because this is the default setting. But because we had a header, all these header strings were handled and put into our data frame header. So we have farm identifiers here. We have farm type as a factor here. We have clade numbers. So these samples, these assemblies are probably the same species because they grouped into the same clade during a phylogenetic check. We have the size here, contact number, and so on, a lot of other things. And also all the antibiotic resistance data later. So what we will do here, and the reason why I wasn't really, I didn't really use this because in some way what we will do now is it can be done in Excel as well. So I don't want to convince you just to everything in R if you already know how to do it in Excel. So if you know that, you can just put the same table in Excel and create a filter on your columns and say, okay, I want to filter my data according to the farm type. I want to filter my data according to size, and I only want size that is greater than something or more than something. But you can also do the same thing here in R. The only reason why maybe it's a good idea to do it here because you can if you find something on your plot that you want to see a bit more clear, then you can just zoom into that thing. I mean, not really zoom in, but filter for that thing a bit more easily. So let's just make a very simple scatter plot first from this data, which is ggplot as the df again, and we are plotting the mean coverage. So this is the coverage value or the average coverage of the assembly and against the GC content of the assembly. And we are doing a scatter plot. That's why it is a G on point. And because we have quite a huge variance in coverage, we have some very high coverage assemblies and some very low coverage. If I'm doing this without a log scale, then it will be much uglier like this. So if you see something like this, then it's of course always good to stretch this small crowded part a bit more, and this can be done easily with a log scale, a log x scale. So now we will use the dplyr filtering functions. So what you see here, this percentage error percentage and percentage error percentage, is almost the same what we will use many times, and we already used a few times in command line. So when I told you in command line that you have command 1, and if you use this, then you can put your output of your command into an output file. If you say output.txt, then everything which will be other ways would put on your screen will be pumped into your txt file. But what you can also do on command line, you can also use this vertical line, which is called pipe, exactly the same as this is also called pipe or pipelining. And you can give the output of your command into your next one. There is a problem with Tom that the gccont object is not found. And you check the table and it is definitely there. So we are here in this line. It has to be some kind of mistyping because the gccont is there. Check again for your variable, your data frame name, and check again for if there is some kind of space there, or this is strange. Okay, I will continue with the piping, but we have to find out the reason. What you can do is just simply put here into your console, say cool names, and the underline df. So with this, you will see exactly what are your column names, and because maybe there is something what you cannot see on your viewer, but it's actually there. And if it's gccont like this, then it has to be good. It only gives this error if there is some kind of mistyping. So if I'm just deleting the T from the end and say this, then object gccont not found. So it's exactly the same error what you get. So that's why I'm thinking that there is some kind of mistyping here or there. But if it's not in your column names, then okay. The header is there now. Perfect. maybe it's not readTelim, or because there are readTelim2 as well. I don't know. Okay, so let's go forward with the piping. So if you are on a command line, and this is what we will do many times in the future, and you have a command 1, and you say vertical line, and say command 2, then command 2 can accept as an input the output of command 1. And this is exactly what is happening here. So I am, by saying sddf, I am actually put the whole data frame to the main output. If I just simply say sddf here, and it will put out the whole data frame. Of course, it's not the whole, because after a few lines, it will stop putting out the data. But if you say just simply this, it will print or put the content of your data frame to the standard output. So this will be piped to a filter, and this is now, we are now deep in the dplyr package. Oh, actually, first go with this one. So now we are filtering to farm type, and only get those lines where the farm type is equals to organic. But we don't stop here, and this is why this dplyr package handling is, especially with these pipes, is very easy, because you are just, it's very easy to see what is happening. Very easy to see that, okay, I have the whole table here, I have a subset of the whole table, which was filtered by this, and then I'm also filtering by coverage, and I'm only looking for those assemblies which has a mean coverage more than 5. And everything which is after this one is exactly the same as we had here. So I'm using the coverage as the X, the GC content on the Y value. I actually made a color. I will tell you what is this color soon. The same logarithmic scale and a different color palette. So if I'm running this, I already have much more information in my picture. I used this color here. What is happening? So what's happening here? Again, this is something what you already know. The substring command is therefore taking a part of a string. So if you go back to your data table, what you see here is CV3 underline 2. That means that it is from CV3. It's farm free, conventional farm free, and it's sample 2. But I don't want to color my dots according to the samples, but I want to color them according to the farm only. So if I'm taking on the first 3 characters from this string, that will always be the farm identifier, because it goes from CV1 to 5 and OG1 to 5. It's always 3 characters. So this is what I'm doing with the substring here. I am taking here substring, I'm taking the farm ID, and I'm taking out from 1 to 3. If you remember, substring is taking 3 main parameters or arguments, the vector you want to extract substring from, the start position and the end position. So I want to extract the first, second, and the third characters. And this will be only CVX or OGX. So this will identify my farm. Of course, we only have OG 1, 2, 3, 4, and 5 here. Why? Because we applied this filter during our process. We don't work with the whole data frame. For ggplot, we usually give the data frame here. Instead of giving the data frame, we gave this thing here, this whole thing, which is data frame filtered by this and filtered by this. So at the end, it's a double filtered data frame, but you can also filter it many other ways. this is maybe a point where it become a bit more powerful than Excel, and also maybe a bit easier. So this will be OG125, and instead of using again the built-in color palette for R, we are using a color brewer color palette. And instead of using scale fill brewer, what we did with the Violin Plot, we are using scale color brewer because we are coloring the dots and not filling the dots. I also made this in a bit different way. So this is exactly the same as you have here. But instead of defining the whole content of the farm type, so if you look at here, this is organic, that's the word which is there. It's actually in a factor. I'm only saying org, but I'm using str detect, which is again exactly the same what we used here. It will give you true or false if this small string part is there. Of course, the org substring is not in the word conventional, so it will give us the same thing. The reason why I put this here, and this is again something you will face with many times in programming, as I mentioned before, almost all programming, what you do, command line or programming, Perl, Python, anything, they are case sensitive. If you don't exactly know that your word is capitalized in the beginning or not, or it's full caps or all caps, then in many cases you can ask the program to do it case insensitive. In this case, you cannot, because in this case, when you say form times has to be equal to organic, you are defining a value. If it's organic with capital O, it won't be true anymore. If I'm running this, I won't have any data because there is nothing which is matching with organic with capital O in the beginning, but there is something with the small. What is happening if I'm doing this here, org, nothing, there won't be anything. But if we go to str detect, I'm pretty sure, maybe not, but I hope that str detect can be case insensitive. It won't be, but I think you can make it case insensitive with regular expression itself. Let me just quickly find it out. If anyone knows, I'm happy to have this information. Okay, we have to put in the pattern we are looking for into this regex as well. So this is what we will do. I almost found, instead of having it here, I just put it here, saying org, but also ignore case true. So if I'm running this, I have all the dots. So that's why it's a little bit better to use pattern search instead of using this kind of clearly stating what you are looking for, because here you can do a ignore case, but of course it has its danger as well sometimes. But in many commands, the grep which we will use, this command grep, there is grep command in R as well, very useful command, but also we will use it a lot of times in command line. You can also make grep case insensitive. In command line, you just say -c. This is how it will become case insensitive. Here, if you grep, again, ignore case here. It is in default false. So in default, the grep command is case sensitive, but you can ask it to be case insensitive. Right. I will stop here, the data management. But I think what you have to memorize from the dplyr is the way how you can very logically build up your chains of filtering and transformations. And as soon as you have this in your mind, you can just go back to your cheat sheet and look at, okay, I want to do this, I want to search for these things, I want to merge these two columns, I want to split that column. So it's very easy, for example, to split this column here. As you can see, I use a semicolon. It's actually not me, it's the program which outputs the taxonomical identification of each assembly. It's the domain, the phylum, the class, order, and then there's the family, genus, and species as well. Later, all of them are separated by semicolons. So if you want to put these specific taxonomical groups one by one into a column, because you just want to look for all the negativicutes or all the spirochetes, then it's very easy to split this column into subcolumns with, let's see, I have this cheat sheet on my desk at the moment, so I can see it. I think it's probably some kind of split function there. Summarize, make new variables. Okay, it's here, but I don't find it at the moment. And I don't want to waste my time and your time, because I want to tell you one more thing. And this is how you will start doing your own scripts. So let me just write this here as comments at the end of this program. So when you start programming, when I start programming, even many times after a lot of years of programming, I just take a paper and write down what I want to do with that data, what I have. So first of all, of course, you have to understand your data. This is not always very obvious, because, for example, you can say, okay, let's do a blast. The blast in default, if you do a blast in a command line, in default, it outputs a very strange formatted output. You can see what is happening on your screen, but you will never be able to, you can, but it's very hard to process that data with a script. So understanding your data is not just understanding the output of BLAST, it's also understanding what kind of outputs you can ask from BLAST. And as soon as you go into the help of BLAST, you will see that, oh, there is actually an option which will give you a tabular output, a simple, very simple table output, where you can also actually define what kind of information you want to see in your table or tabular output. But for that, you have to understand how BLAST works. I mean, not how really BLAST works, but how you can use BLAST. So it's very important to know what your data consists of, what are the delimiters, what are the different fields, what you want to process. And then just simply draw like a scheme, right, or like a flow chart of the way you yourself would do the processing. If I don't give you a computer, if I don't give you any calculators, I just give you the big table printed on paper, what would you do? How would you go through? Okay, I check the first line and I'm looking for this field, and if this field is what I want to see in that field, then I will take the other cell and multiply that with the third one. Okay, so that just make a simple flowchart, because you will do exactly the same in your program. You don't have to think differently. You will do the same. You will just have to find those functions, those building blocks, what you will use to do this exactly the same thing. Okay, the next thing is a combination of, especially if you are doing it in R, combination of sheets and Google and your previous scripts. I'm in a bit of advantage here because I have much more scripts than you have, but of course I also started somewhere. For me, many times it's just going back. Oh yeah, I remember that was the project where I made the same kind of graph and the same kind of data processing. Let's go back there, copy and paste, and then I have the same thing. But this is what you will also have as soon as you start making your own scripts. But until you don't have, you just go to the cheat sheets and go to Google and ask the same, very simply the question: how can I check the column GC content value in each rows in a data frame, or something like this? And there will be a solution, and there will be maybe more than one solutions, and then you can just find the one which you see the most simple, or which you already met with, and then use it. But before you use that command or use that package or use that function, please always read the help of the function. You don't have to read the help of a function if you are using it more like already 20 times you used. But if it's new for you, read the function. If you read, always be very cautious on what are the default parameters, what are the input parameters. So for here we have grep, you know that you have to give pattern, you have to give X, and you can also give a few other switches as parameters. Read what is that. So if your pattern is a character string, okay, so it is a string, it is a sentence or anything, or a sequence. And X is what you want to look for in your, no, it's the other way. So you are looking for a pattern in a text with grep. And also always look for one more thing, not the details. There are some interesting things in details, but the other thing which is very important is the value. So each function is taking inputs as arguments and giving back a value, and always understand what it gives back. It's usually very simple. With grep, it will give back that line or that piece of your vector, which is matching your pattern. But sometimes it's a bit more complicated. Sometimes it's a special type of variable, and then you have to understand how can I. So if for example, there is a function which gives you a list, which is different from data frame, it's a different variable type, but you want to use ggplot, and ggplot likes data frames more, and maybe it can automatically, I think, convert a data frame, but maybe it's not converting in that way as you would like to convert it. Then you have to go to Google and type in how to convert a list to data frame in R. And then you will understand how you can do that one extra step when you are converting your data.